I'm Hazel Sive. I'm professor of biology at MIT, and I'm a member of the Whitehead Institute for Biomedical Research. The thing that I study is how a single cell becomes something that's got lots of cells like me or like you. I know in your class, Eric has introduced you to the concept of cells, and the thing that has fascinated me since I was an undergraduate is how a single cell, the fertilized egg, can turn into something really complex. I started my career in South Africa. That's where I was born, and that's where I did my education up to the end of my undergraduate work. And then I came to England, where I taught school. And then after that, I came to the United States, where I went to graduate school and eventually landed up a professor here at MIT. What I want to tell you a little about is the things that we really study. What I told you about this whole question of single cell to animal or plant with lots of different cells, lots of different kinds of cells, or working together to make a human or a fish or a tree. That's the really big question that we think about. In my research group, we think about just a part of that question. And one of the things we think about is how the brain forms and how the brain gets its particular shape. Your brain started off as a long tube, and that long tube had different kinds of cells along its length, but then it folded up, and it folded to make a nice round compact kind of structure that packs into the skull, and that's a safe, secure place for your brain to be. One of the things we study in my lab is how the brain folds up into this compact shape that gives security and function to the brain. And one of the particular things we study is how the sheet of cells or the tube of cells that makes the brain folds in a particular way. A while ago, an undergraduate in my lab discovered a new kind of way that cell sheets, big sheets of cells, that's what the name implies, all stuck together, fold up. And this kind of folding is called basal constriction. And you're going to look at a movie that shows this basal constriction happening in a particular part of the brain. Now, we can't study these things in human brains, of course, but we can use models, animals that we can study to understand how the brain gets its shape. And we use the zebrafish to look at the brain, and I'll show you some zebrafish in our lab in a little bit. And you're going to be looking at a movie where the cells, every cell is outlined in green by a protein called green fluorescent protein, and where the nuclei of the cells, where the DNA lives, are in red. And you're going to be looking at the different shapes of the cells as the brain forms. And you can look through the movie and see the particular cells undergoing their basal constriction. It's a very beautiful movie made by Isabel Brackman, a postdoc who works with me in our research group. Doing research is something that I love. It's the hardest thing I do, and it's the best part of my day. It's a great part of my day because I work with students at all levels, sometimes high school students, undergraduates, graduate students, and postdoctoral fellows who all contribute to how we discover things in the lab. We work together as colleagues. They come up with ideas, I come up with ideas, we discuss them together, we tell each other how wrong we are, and then we come up with new ideas that might be even better and maybe ways to help us explain how life is put together. In our case, how the brain gets its particular form. The whole process is really friendly, but very serious, and we work very hard. We work with whole embryos. It's a real privilege to work with these animals, and we take care of them very carefully. And we work using microscopes and the tools of molecular biology that Eric has introduced you to in the course. We use recombinant DNA methods, we use transcription, we use translation, and we use microscopy a great deal to look at the actual cells in the embryo as the embryo is forming. It's a really big enterprise that we all put our hearts and souls into as we're doing our research. MIT is one of the greatest places in the world to do research, and every day I come in here, I feel really privileged to be able to do the work in this place with my wonderful students and with my wonderful colleagues who are also professors. It's just a fantastic place to work. And I'm happy now to take you into the lab and show you some of the things that we actually work with. This is our microinjection room. This is where we take our little tiny zebrafish embryos that are about 0.2 millimeters in diameter, and we inject the very early embryos with various substances. In most cases, we're trying to knock down gene function. Sometimes we increase gene function. And then later on, we'll look to see what's happened to the brains of these embryos and whether or not they've developed normally. The thing that is really cool about zebrafish embryos is that they are transparent, and you can look right inside them. You can see the cells that are developing and dividing and becoming different kinds of cells just by looking at the embryo directly. And that is really, really amazing. This is Dr. Jasmine McCammon, one of the researchers in our lab, and what she is doing is putting a very tiny injection needle into something called a micromanipulator. The needle is a glass tube that we've drawn the tip out to, so it's about 5 micrometers in diameter. And then she's placing this carefully so that she can look through the microscope and inject her embryos by very gently moving the needle up and down. This apparatus is a microinjector that allows her to give tiny amounts of fluid, about 1 nanoliter, to each of the embryos. And that nanoliter of fluid contains DNA or RNA or protein, depending on what we're injecting into the embryo. We can get hundreds of embryos every day that are injected, and so we can do experiments that involve very statistically significant numbers of embryos to get data that is really valid and allows us to understand some truly profound things about what it takes for the brain to develop normally. What Dr. McCammon is in doing here is not only studying how the brain functions normally, but how it develops abnormally in cases, for example, equivalent to autism or schizophrenia in humans. And so she is studying genes that are associated with human mental health disorders that may make the brain develop abnormally. And we're using the zebrafish as a tool to try to understand something about human mental health disorders.